thank you, everyone. Welcome to those of you who are viewing this. We wish we could be there with you, and we want to thank you for taking time to engage on this topic, which everyone on this panel, and that includes myself as the moderator, feels very, very strongly about. My name is David Cameron. I am the Global Head of Novel Trial Design at IQVIA, where we focus on the application of novel trial designs where appropriate to impact several aspects of clinical research development, which we will be talking about today. I will shortly introduce our panelists, and then we will walk through a series of questions that we hope will be relevant to you all. And we wish we could take questions, but unfortunately, the format does not allow for that. But perhaps if there's anything that is of really burning desire for follow-up, you can reach out to us through the contacts that you have at the conference. And I also, of course, want to thank the Meeting on the Mesa folks for giving us this opportunity. So let me just briefly introduce the topic. It's been very apparent that the use of innovative trial design and execution is on the upswing. That is driven not just by our current ongoing situation with respect to the pandemic's impact on clinical research, though that has certainly highlighted several areas that already existed in terms of challenges in clinical research, but also the unique characteristics of cell and gene therapy clinical planning and execution. And we want to try to cover in our content today, not only what we've learned from the COVID-19 pandemic, but also what are the unique applications and challenges and solutions that novel trial design brings to cell and gene therapy. As a panel, we've spent some time talking about what we thought thematically would be most relevant for this topic, and we'll spend the session addressing these themes through the questions. The three themes that we settled on were, first of all, design and execution efficiency. And that's from multiple perspectives, not the least of which is the patient's perspective, their ability to get access to medications quickly and safely, and the most relevant treatments, of course. The second theme is really the idea that design innovations encourage collaboration across stakeholders and across clinical plan development transitions. So you'll hear about how patient advocacy and payers and product manufacturers and investigators all can come together through the use of novel trial designs to drive clinical development from hypothesis generation through perhaps natural history of disease studies all the way through hypothesis confirmation in, say, master protocol and basket trials. So that concept of collaboration, both sort of vertically and horizontally, is something else that we'll touch on. The third theme I've already mentioned earlier, which is what have we learned about the innovations that have been necessitated by the impact of COVID-19 on clinical research. Those have highlighted, they've certainly been new challenges, but I think there's also been some highlighting of existing challenges, maybe exacerbating of some existing challenges. And we want to give you an idea of how those learnings will carry forward as we go. So with me today, I have four distinguished panelists, and I will just briefly introduce them, and then we'll get right into the questions. Our first panelist is Dr. Sabrina Paganoni. She is a physician investigator at the Healy and AMG Center for ALS, amyotrophic lateral sclerosis, at Mass General in Boston. She's also co-director of the Neurological Research Institute at Mass General and an assistant professor at Harvard Medical School. Dr. Paganoni's research focuses on clinical trials and therapy development for ALS. She is a co-principal investigator of the Healy ALS platform trial, which is a master protocol trial, novel trial design, the first such trial for ALS in the world. And as a principal investigator of several ALS clinical research projects, she has been using novel trial designs as well as novel operational paradigms through telemedicine and digital technology. Her research has been funded by the NIH, several ALS foundations and nonprofits, as well as industry. Next, we have Dr. Shaji Kumar, who is a consultant in the Division of Hematology and professor of medicine at Mayo Clinic Cancer Center in Rochester, Minnesota, where he also serves as the medical director for the Cancer Center Clinical Research Office, which oversees interventional trials across the 3 locations of Mayo Clinic Cancer Center. He's also vice chair for research in the Department of Medicine at the Mayo Clinic. He received his medical degree from the All India Institute of Medical Science in New Delhi. And Dr. Kumar's research, which has been presented internationally and in several peer-reviewed journals, focuses on the development of novel drugs for the treatment of myeloma. And he is the principal investigator of clinical trials exploring new drugs and combinations for newly diagnosed and relapsed myeloma. In addition to several other prominent committees on which he serves, he is the co-chair of the NCI Myeloma Steering Committee. Our next panelist is Dr. Han Cho, an associate professor of medicine at the Icahn School of Medicine at Mount Sinai. He's an attending physician with the Multiple Myeloma Service at the Mount Sinai Tisch Cancer Institute and serves as chief medical officer at the Multiple Myeloma Research Foundation, the MMRF, which you will hear us talk about that as well going forward, where as part of the MMRF executive leadership team, he develops clinical research strategy and oversees the clinical development program. Dr. Cho's laboratory is investigating novel therapies for multiple myeloma in several areas. He also leads the Multiple Myeloma Research Consortium, which is a group of 23 leading research centers dedicated to researching and advancing treatment options for multiple myeloma patients. Dr. Cho is one of the principal investigators for the newly launched MMRF Cure Cloud, which is an innovative research study that includes free at-home genomic testing to inform treatment plans and accelerate the application of precision medicine for these patients. And our next panelist is Dr. Eamon Shalaby, who has over 20 years experience in global development and commercialization of new medicines for patients with cancer. He currently leads the global development program for an adaptive cell therapy targeting solid tumors, an adoptive cell therapy targeting solid tumors at GSK that has achieved FDA breakthrough and EMA prime designations. Previously, Dr. Shalaby was the global vice president in AstraZeneca, leading the development team for durvalumab, and then he led the global medical affairs program to support its pre-launch. Dr. Shalaby is a leading advocate for external clinical academic collaborations, again, one of the other themes that we're focused on, and previously contracted with the Cancer Research Institute, the Canadian Cancer Trials Group, and the Harvard Business School to drive philanthropic support for academic clinical innovations. Prior to his work in the pharmaceutical industry, Dr. Shalaby also had a successful career at the National Cancer Institute, and his degrees include a Doctor of Pharmacy from USC and an MBA from Johns Hopkins. So with that, I thought we would start touching on the themes, and we'll start with a question from, for Dr. Paganoni. Dr. Paganoni, we've talked a lot in our discussions prior to this session about how innovative trial designs, things such as master protocols, adaptive designs, maybe the use of real-world data or hybrid real-world study designs, how they can impact the efficiency and quality of clinical research, by which we mean really bringing more relevant treatments to patients sooner and more efficiently, and also expanding patient access to trials. And I know from your work in ALS that this is a set of objectives that is near and dear to your heart, and I was wondering if you wouldn't take us through a little bit how you've seen these designs work and how they address objectives such as those.
Absolutely. So I want to start by sharing our experience. As you said, we recently launched a master protocol for ALS, or Lou Gehrig's disease, known as the Healy ALS platform trial, and the enrollment is going really fantastic. So really this is I guess now we know that really what we have been planning for the last year is really what patients wanted and something that can be incredibly effective. So the motivation really for us came from the patients. As you may know, ALS is a rapidly progressive neurodegenerative disease, and most patients die within 2 to 3 years after onset. So obviously for them, the speed and the efficiency of the clinical trial development landscape is incredibly important. They essentially have one shot at a clinical trial, or maybe they can participate in a couple of trials, but it's incredibly important for them the trials and run and executed really efficiently and quickly. So this was really the motivation. In ALS, we had this unique situation that now we share with other fields of medicine. That is that we have a lot of drugs in development, a lot of different investigational products essentially waiting to be tested in a rare disease population. So a couple of years ago, we sort of came together as an ALS community, mostly the academic ALS community that has excellent ties and collaboration with industry. And we said, okay, how do we rethink the landscape? How can we really match this huge patient interest and motivation and desire to participate and the growing pipeline? How can we really rethink the way we are organized so that we can really test as many treatments as possible rapidly and with patient access? So this really was the motivation. And interestingly, we turned to our colleagues in cancer. So I'm not surprised I'm the only non-cancer specialist on this panel, because really the concept of master protocols started in cancer a few years ago. And now it's growing in the neurosciences as well. But I think we really wanted to learn from people who were experienced in this field and with this approach. And so we learned from many different groups. I have to say that there is a nice community of people who work with master protocols, both in academia and in industry, and they've been very generous and really shared all their knowledge and experience. And so we spent a few months setting up this new trial. And so basically, we were able to match, sort of, create a series of collaborations with different industry partners that wanted to participate in the master protocol. We worked a lot with the patient community. As an academic organization, we have a network, we collaborate with a network of academic centers. We all came together, many experts in ALS contributed their knowledge so that we could really build what we think is an optimized protocol for testing drugs in ALS. And so we built what we think is an ideal protocol at this stage of ALS science. It's a master protocol. We can add many more treatments. We're already, adding more. We launched with 3. We're about to add number 4, in the next few months, and then next year we should be able to add another 3 drugs. So essentially, and we also built the whole infrastructure to increase access from patients. So I think all of this work is now paying off, and we see it with the enrollment numbers. So I'll just stop there, so to give others an opportunity to weigh in. But I have to say, as a person who works in the neurosciences, we're certainly grateful to our colleagues from cancer who really paved the way, and now we are benefiting from that.
Great, thank you. Anyone else on the panel want to add anything to that response? And feel free. I know it's the logistics are difficult with the video conference, but we'll moderate it.
So maybe I can just add on to that in the sense of, I think in the cancer, especially with the rare cancers like what me and Dr. Cho deal with, which is multiple myeloma, again, not very common compared to the more common solid tumors. We kind of face the same issue, right? We can't really do multiple large trials to demonstrate the value of individual drugs, and especially with there's really an explosion of new drugs out there, which is a good thing, and it is clearly based on the understanding of the cancer biology. But and many of them appear to be quite attractive and potentially useful in the rarer cancers. And so and Han can probably touch upon this further too, is that we had in the MMRC developed a master protocol, which is called the MyDrug trial. It's again, myeloma drug regimens using genomics. So our goal was to basically screen these patients for a bunch of mutations, which we know from a large experience. It's roughly about 10 or so, 12 mutations that are commonly seen. So screen the patients, identify the mutations, treat them with a drug that's specifically targeted towards that. And it's basically a multi-arm protocol. But unlike maybe not as much like the neurological disorders in cancer, especially something like myeloma, we know that going after just a target, which may be in a proportion of cells, may not necessarily be the end all. So we have to really improvise on, kind of build on that design. So we designed this trial to have just not looking at the targeted agent, but also adding a commonly used backbone to it. So I think, we had this whole window design where we would treat the patients with just that targeted drug to see if it really works. There's a proof of principle, and then follow that up with a combination strategy so that whatever response we do see kind of sticks and provide a more durable benefit. So clearly the whole concept of these multiple small arms that is, very nimble trial where you get an early read, is it working? If not, let's replace it with something else that could potentially be useful. Concept I think is really catching on.
I think, let me just chime in here. I think Shaji is being too modest. He was one of the architects of the MyDrug protocol. But it is a very good example of how we can use cutting edge technology in the setting of a rare cancer to rapidly deploy multiple different agents, either tailored to a specific setting, a specific mutation, or there are various permutations of the sort of platform or basket or umbrella trial model in which you can the goal would be to hypothesis test application of different types of agents, different classes of agents for specific either rare conditions or even subpopulations of rare conditions in a way that would give you results quickly, allow you to move on to advanced phase testing for the winners, and move on from the ones that do not appear to be effective in that setting. And I think that's a really important concept, which, as was pointed out by other panelists, was deployed in cancer, but has now spread into other areas as well. And I think one area where this has not really been explored yet, which potentially could have a great impact, is in pediatric cancers, which by definition are rare cancers. So I think there's a lot of opportunity there, which these types of innovative trial designs may be of great benefit.
Thank you very much, Dr. Shalaby. Did you want to add anything on that question before we move on?
Yeah, no, just to congratulate Dr. Paganoni for getting this off the ground. You mentioned it took us a few months to get it off the ground. Be interested to learn from you, from the oncology space, how you could do it in a few months. It takes us a bit longer. But what I will say is really, congratulations for moving this into other areas. There's some operational learnings, some key design issue learnings. But one of the things I just wanted to add is, the type of team you have to assemble to get this done. And I don't know if yours is a multi-site protocol or a single-site protocol, but there's been a lot of lessons learned along the way, especially, as we kind of jump into these study designs and with the kind of the key theme of execution efficiency, right? Sometimes you don't know what you don't know until you really get into it. Whether it's the regulatory pieces, the clinical operations pieces, the statistical considerations, the negotiations with the different companies. Yeah, there's certainly kind of a series of challenges to address along the way. But I'd just be curious, if you had to say, what is the one big learning in going through this experience for the audience, and just curious, where would you put your finger on? Hey, I learned this along the way.
I would say that it takes a village, and that you want to partner with the best people, the most experienced people. So I mean, it's kind of obvious, but in ALS we had a little bit of a unique and privileged situation because academic centers have been working together for 25 years, running individual trials. So we have a network, there is a network of academic centers, essentially many of the major ALS clinics in the country. They have been working as a network, a collaborative research network for many years, including several trial-ready sites that have been working on different trials, both investigator-initiated and industry-sponsored. So that's sort of the background. And as an academic group, we developed over time, I should say I'm a more recent member, but our leaders and colleagues have built over the last 2 decades, essentially an academic CRO, or kind of an academic group that had the capability to not only design but also execute clinical trials. That includes project management, data management, biostatistics, outcomes training, monitoring, etc. So it's essentially a completely independent group that can collaborate with others, but has all the functionalities within the academic environment. Obviously, there has been, the group has executed individual trials until more recently when we started working on the platform trial. And so we spent some time really learning from people from cancer, learning their challenges, also on the operational aspect. I would say that you're absolutely right. The operations, so running a master protocol is not just collating together a bunch of different trials. You really need to rethink the entire infrastructure and the entire operation. And that's true for the contracts, if you have multiple industry partners. That's true for the regulators, because you do want to engage with the FDA, before you even launch the trial, right? To understand how to really report on safety and all the other regulatory pieces, as well as the database. You need firewalls, you need processes to be able to report on the master protocol as well as the individual trials. So it's very complex, but we were fortunate, and the reason I think we were able to go so fast is because we have had that experience and our in-house processes that needed to be adapted. And fortunately, we were able again to learn from other people that have done it successfully, and so that's why we could launch very rapidly. But we do have a large dedicated team really working on this full time.
Thank you.
Yeah, thank you. Just a quick follow-up question for those of you that have been involved in the master protocol trials. Are there any, have you incorporated any adaptive methods to that? Even simple adaptive methods like sample size re-estimation or group sequential designs where you're looking for that futility stop. Have any of these projects incorporated those more biostatistical elements? Maybe Dr. Paganoni, we'll start with you on that.
Sure. Yes. So when we talk to people who have done, other adaptive trials, they told us to start, with sort of simple adaptations and then build them over time. And that was a good suggestion for our specific field. We don't have as many sort of molecular biomarkers as maybe the cancer world has, and so they can do other things. But for us, basically the adaptive features of the trial are that we can continue to accommodate new investigational products, so we can continue to add regimens to the master protocol, and we do have futility stopping built in. We have optional early stopping for efficacy that you can use or not for a particular investigational product. So those are the adaptive features that we are using at this stage. Now, other things that are more complex, like responsive adaptive randomization, is just not a good fit for our specific science, the state of the science in our field. But obviously, my colleagues from cancer might have a different view on that, given the state of the science there.
Dr. Kumar, Dr. Cho, anything to add on that?
No, I think it's, some of the designs that we have built into, for example, the MyDrug trial, we do have, the ability to look early on to see if this is something that is worth pursuing after the first few patients are on. But I think I just want to point out some of the specific challenges from an oncology standpoint too. For example, the MyDrug trial, it ends up being a little bit more complicated than many of the other platform trials out there, because it does incorporate drug combinations, which also entails for us to do a dose escalation part kind of mixed into that platform. So it ended up being quite a multi-dimensional from multiple different standpoints. So incorporating the combinations, the dose modifications, and also building in the futility design to make sure we do call it off early on is certainly there. And then we certainly encountered some things which we didn't bargain for. So for example, we had run into some potential for toxicity with one of the drugs, which ended up with the FDA telling us you have to do a randomized component to that. So then now we have another clinical trial element of randomization that we actually incorporated into the trial design. But I think kind of going back to what Dr. Paganoni was talking about last time in terms of lessons learned, I think the key thing is, you have to have a group of people who believe that this is the right thing to do. You have to have a question that you all feel very strongly needs to be answered. Particularly in the my drug trials setting, we knew there were a group of patients who don't do very well, which is what we call the functional high-risk group of patients who are not benefited by the current therapy. So clearly there's a need. And then we have the tools, which is obviously the genomic testing. A lot of that data had been generated by the Myeloma Research Foundation itself. That was the basis of the study. And then finally, I think getting all the pieces together, I think the regulatory pieces is a challenge. Having the infrastructure built up is a challenge. Negotiating with multiple companies is a challenge, but all can be overcome as long as you know that you are on the right path. And Hunt has been involved a lot more with logistics too. So.
Well, I think Shaji really hit a lot of the high points, but what it does point out is that it is feasible. It is feasible to assemble a coalition of the willing, and that includes not only academic investigators such as Shaji, non-for-profit organizations such as the MMRF, but even the pharma companies. The fact that we were able to negotiate with 6 or 7 pharma partners in order to execute this, and that we have had successful ongoing negotiations adding additional pharma partners as we bring new cassettes into the trial, is a testament to the will to answer these very important clinical questions. And using this type of innovative design, even though there was challenges, with purely practical matters such as contracting and operational matters such as drug delivery and so forth, we were able to, this was, it's feasible. It can be done. And I think that shows that it can be done here. It can be done elsewhere.
If I can add one more thing, I think you're absolutely correct. I completely agree with everything you said, and it can be done. And I think it's actually advantageous for all parties involved. So I think that's why over time people tend to buy into this, because why would you do things differently when you see the advantages of it? So I think as more master protocols are developed and implemented and have successful results, people will see that there's something to be gained from participating. So, and we can get more into that, but I think, there's a lot of efficiencies in terms of sample size, data, biomarkers that can be generated for both industry and the scientific community.
Thank you. Let's move on to our second sort of question theme. And Dr. Kumar, maybe you could kick us off. And this one is really more along the lines of, the necessities of innovation in this age of impacts to clinical research as a result of the pandemic, where among other things, we've learned that access to patients and patient populations, but also to sites that conduct research it's finite and precious. And there have been other learnings too. I'm curious from your perspective, what you've seen, what you think might carry forward, or what we might have learned that's practical and constructive as we move forward beyond the stage we're in.
Absolutely. No, I think the COVID clearly made us all put our thinking caps on for sure. At least I can speak from the perspective of what happened within the clinic here. As soon as, obviously when the pandemic started, a lot of things just went into a shutdown mode. But then we kind of regrouped and figured out how can we continue to do these clinical trials. And I think particularly for the cancer patients where the clinical investigation is pretty much embedded within the clinical practice, it's really difficult to just do one and not do the other. So we really had to, on the fly, figure out how to continue with the clinical trials. So that we did, incorporate a lot of changes over the initial weeks, which I think some of which are things that we have talked about in the past as methods to make clinical trials more efficient. And this really accelerated the process and at least temporarily gave us the regulatory permissions that were needed and which we hope will stay long term. So first thing is consenting. It kind of kicked off, I wouldn't say kicked off, at least accelerated an ongoing project looking at electronic consenting. So we are certainly kind of moving now into a completely electronic consenting mode, where depending upon the risk involved in the trial, maybe still have the patients be in the clinic, but maybe using an iPad type device. But importantly, maybe once that initial consultation is done, patients could even sign the consents remotely through in the setting of a video visit and so forth. In terms of enrollment, we've kind of put a hold on enrollment right away, but then gradually opened them up. I think we felt more comfortable opening up the phase 3 trials and gradually getting on to the phase 1 trials where the benefit may not be that apparent as in later phase trials. Again, the intent at that time was to try and reduce the travel to the healthcare facility and decrease the risk of exposure and potential for infection. But I think this is, again, I think kind of tied in with the whole consenting process, the whole consenting, enrollment, the accrual. There are a lot of elements of that that can really be done remotely, which again kind of brings up the whole concept of telemedicine, which I think is probably the most important takeaway from the whole pandemic situation, whether it be clinical practice or clinical trials. There's a lot of things that don't need the patients to make that trip to the clinic. So we have tried to change protocols, or at least have temporary deviations from the protocol to allow patients to have video visits instead of coming to the clinic. Patients who need blood work done in between, sometimes we would send a kit out to them, have them go out to the nearest small clinic that they can go to, get the blood drawn and send it back to us so that we still have the results that we need. Certainly we did end up not doing some of those PK or purely research type studies for that initial part. Obviously that is not something we want to do long term. We want to make sure we can do that. So the question is, how do we attain that going forward? And one of the options obviously is kind of taking the clinical trials to the patients, right? I think that's what the whole telemedicine does. In addition to the visits at home, can we even have research personnel maybe going out to patients' homes, maybe doing some limited assessments, getting the blood samples, and that could not only, I think, increase the attractiveness for participation from a patient standpoint, but clearly, certainly help us with accrual. We also talked about, we also did a bunch of changes with drug distribution. So a lot of the oral drugs that patients were on in clinical trials, we were able to ship them directly to the patient instead of them, having to come back every cycle. We would do some of the remote labs and then deliver the drug to the patient at home. Obviously, that's not something we can do with the IV or parenteral drugs, but certainly flexibility that's afforded by the oral drugs. We had people do imaging studies at home and send the films over so that we can kind of compare them at the enrolling institution. And I think there are a lot of what was really striking was the flexibility from the IRB, and they really did allow us to make deviate to some extent from the protocol as long as the patient safety was not compromised, but also allowing us to kind of batch together some of those deviations to be submitted together so that the regulatory burden doesn't become too much. We incorporated a lot of, improvements within the EPIC, creating flow sheet tools to kind of keep track of these deviations so that it becomes easier towards the end when we have to report them to the IRB. So, so a lot of different changes. Certainly, we hope, especially the telemedicine aspect of it, would stay going forward. But certainly made us think through the clinical trial process.
Yeah, we had a very similar experience. So we actually had the investigator meeting for the trial to launch the trial. We were ready to launch in March. So the investigator meeting was on March 12th in Boston. Now, the first COVID cases in Boston happened exactly 10 days before. So we got a message 5 days before the investigator meeting was scheduled to happen. We had, hundreds of people coming into Boston for that. We got notified that we couldn't hold it because there was basically a hold on those in-person meetings. So in over a 5-day period, we were able to turn the entire investigator meeting to virtual. So we did hold the investigator meeting on March 12th and 13th in Boston. We couldn't start enrollment right then, but that's when we started to see the amazing efficiencies of a master protocol structure, because essentially, so think about this, we were gonna launch the platform trial in March for 3 treatments. under the same master protocol. So we made all the changes that Dr. Kumar explained, same thing, home blood draws, telemedicine, home nursing, all those changes so that, some of the procedures could be done remotely. We were able to change the entire protocol, 3 drugs, in one IRB submission, essentially. So, so had we had, if these were 3 separate trials, we would have had to do it for each of them separately. Instead, this was a master protocol. So the framework that's very similar to what you discussed in terms of adaptations was, applied to all of them at the same time. And so we were able to launch the trial then, and we actually enrolled the first participants in July because we had, what took the most time was really more the contracts, to make sure that we could actually implement these changes more than the protocol or IRB or FDA process, really. So we had a small delay. Nevertheless, we were able to launch 54 sites, the sites are being activated, all of them now, but essentially that allowed us to improve the protocol because now we have more flexibility. And so we do hope that all of those adaptations will stay because they make life easier for everyone. And also, should the pandemic recur or get worse in whatever location, I mean, we are present in many states, we are able to use some of the optional telemedicine visits or other things that can be turned on and off depending on the need now or in the future. So I think in a way, having one single protocol helped us make those changes faster for all the investigational products.
Great, thank you. Let's keep moving just in the interest of time to make sure we cover our last 2 questions. Dr. Cho, the theme of collaboration, the theme of how you move through the entire clinical development plan is something that that we've discussed amongst us here that I think would also be of interest to our viewers. Do you want to share your perspectives on that, especially given your unique position with the MMRF?
Sure. So I think we've talked a lot about my drug, but I think it's a good example of how this process can be implemented. And it does require collaboration at many levels. So for example, the protocols and the sub-protocols for the specific agents used in this trial were drafted amongst a number of academic investigators led by Shaji in this case. And there was all the associated design aspects of it, such as the biostatistics and so forth, were accomplished on a collaborative basis. And then obviously we had to have a buy-in from our pharmaceutical partners to commit to the overall design of the master protocol, agree on the screening tools that we were using in this case, using a novel genomics platform to identify actionable mutations or overexpression, and then to agree to a common backbone therapy and implementation of the sub-protocols as equivalent but not comparator arms within the protocol itself. So it requires intellectual collaboration. It requires operational collaboration. So this was headquartered at the MMRF, but required interactions with central IRB, with institutional IRBs, with the FDA, because the MMRC, the not-for-profit, is the IND holder in this case. We're directly managing a CRO, and that is another form of collaboration that is necessary to carry out a trial of this scope. And then, of course, our open communications with our pharmaceutical partners and fostering that horizontal integration of all of the stakeholders in the project in order to keep it going, running smoothly. For example, we're putting in the first major amendment for this protocol is going in, where there's going to be operational changes that were made in agreement with the FDA based on what we've learned with the first 30 or 40 patients coming in. We are altering one of the sub-protocols, as Dr. Kumar mentioned, to include a randomization step, and we're adding 2 new arms, so adding 2 new pharmaceutical partners and their agents into the mix here. And so this type of, the fact that it's feasible and that we can do this is due to the collaborative nature amongst different stakeholders in both in the academia space, pharma, and as us as the not-for-profit foundation running the running and financing a substantial portion of this clinical trial. So this is just one example, and depending on the type of trial, there may be other types of collaboration that could be involved. For example, just to carry over the theme of what COVID has taught us, a lot of the innovations that Dr. Kumar were talking about, telemedicine and remote labs and those types of factors that could make clinical trials more palatable to patients and more easy to implement across larger health systems had been talked about for a long time, but the crisis showed us that it's feasible, right? So that actually opens up the door for a lot of other stuff that has been under investigation or hypothesized. For example, using wearable devices as monitoring within the scope of a clinical trial. So you could potentially design a trial where you would incorporate a technology partner that had wearable technology and the reportage of that, and including that within the trial design itself. And another aspect of this, which may be a little bit further down the road, but which has now been pushed forward, is hospital at home models. So home care models have been in development. You know, Medicare has put a lot of money into these hospital at home models in different healthcare systems. Others have developed their own virtual hospital models, virtual clinic models, where medical personnel, including physicians, go to the patient's homes, can do physical exams, can do blood draws. And if that is a mechanism to deploy within a clinical trial to make it even more feasible to deliver clinical trial care without requiring the clinic visits and so forth, that's another set of collaborations that could potentially be incorporated into future trial design. So these are really radical departures from conventional or historic trial design, but ones that could be implemented to make clinical trials more efficient, accrue faster, more palatable to patients, and more rapidly answer the important questions that we're trying to address with these clinical trials. And you can see how these collaborations can extend far beyond the sort of doctor, patient, pharma company model to include new technologies, new practice models, and new ways to deliver clinical trial care to patient populations.
Dr. Cho, maybe if I could just jump in here for a second. This is an area near and dear to my heart, as you know, and the power of this collaboration is I think still untapped. I think we're just really at the beginning of watching this space grow. MMRF is certainly leading the field in many, many ways, but you know, just to put an industry lens on it for a minute, there's nobody closer to the patient than you and your group, right? You know holistically kind of what patient needs are, what the issues are, what the challenges are, and you know, typically we get you know, one group at a time, different academic centers, different feedback, advisory boards, and we even have patients on some of our review committees. We have a team dedicated to patient groups. But the depth that you guys go into holistically to understanding what the patient needs are across all of the stakeholders really provides a lot of value into what are the unmet needs beyond just a core development trial. So the first point I just want to highlight is tapping into that knowledge of what the patient needs are and incorporating those into clinical trial questions. But also, from an industry perspective, we're not going to be able to do all of the trials that are needed to answer all these questions. And so the opportunity to fill those evidence gaps in the patient populations we can't enroll in, or the combinations that we maybe not have access to that you do, or some of the, you know, innovative technologies is really, really a key, a positive value kind of merit partnership, if you will. I think where it gets a little challenging is when we try to compete with each other for core drug development that's designed for regulatory purposes and for, you know, potentially drug approvals. However, with the caveat is that we know that there's been several drugs that have been approved based on, you know, nonprofit patient groups, and people probably already aware, but you know, Tarceva was first approved based on a collaborative group model. Avastin in lung cancer was approved based on a collaborative group academic-led study. There's a lot of precedence for successful regulatory outcomes, especially in the adjuvant setting, but that's where it gets a little bit tricky. But I will say that we're just really tapping the beginning part of these collaborations. I really hope for much more success over the years for these collaborative models.
Thank you. Excellent. Well, why don't we move to our last question, just in the interest of time, and then we can come back to some of these topics at the end if anyone has anything to add. You know, coming back to you, Dr. Shalaby, you know, just to bring it to the relevance to cell and gene therapies, you know, the novelty and the innovation that we've seen. You know, what are some of the challenges that occur in cell and gene therapy research that, you know, innovation can help with, and maybe help us move the clinical research productivity forward?
So how much time do we have, David? There's no lack of challenges, right? Certainly. So let me, if I could just kind of raise the two big points up front, and maybe take the others offline as needed. But if there's two big things I would suggest to take away. One is we're doing a traditional one drug, one protocol, one question, one answer, like we've always done, should be the exception. I think every single cell therapy should go into a master protocol or be a part of a master protocol. And I'll come back to that in a minute. And the second point is that we should assume that we cannot do a randomized trial with a cell therapy. The exception is that we can do a randomized trial eventually for a cell therapy, but the base case should be doing a single arm trial, looking for a big change in the standard of care with a, you know, big delta in the risk benefit in a positive direction, and to do some sort of real world control arm or synthetic control arm to support that big benefit. So those are two big assumptions that we deal with kind of in the cell therapy world. So if I can just build on the first one a little bit, for cell therapy, we typically don't strike it. We typically don't win with the first in generation, if you will, of the cells. We're learning as we go. We're engineering new technologies into that same cell platform or additional, you know, whether it's opportunities to enhance proliferations, decrease cell exhaustion, penetrate into tumor microenvironment, or incorporate, you know, learning from the cell phenotypes in the manufacturing process, and then iteratively learning from generation 1 to generation 2 to generation 3 and 4. And so to do that in separate protocols just inserts too much white space in the development paradigm. So what we're doing now is we're developing these what's called parent-child IND development strategies, where you actually have the first generation cell and the second and third and fourth generation cells that fall under kind of an umbrella IND, and all of those drop into the same master protocol. So as we learn from each cell, we can then build onto another cohort in the next cell and the next cell and the next cell, and hopefully one of them will work really, really well. So that's kind of the parent-child IND strategy. So anybody who's doing one cell, one protocol, one question, one answer, I think should be looking at these innovative trial designs and thinking differently. And again, I'm still trying to find the person who can help us figure out how to do a randomized trial with an autologous cell therapy, right? It's not an easy task. It is, I don't know exactly how to design it. Somebody probably way smarter than I am, or my team can figure that out, maybe in the liquid tumor space, but I challenge the solid tumor space to figure this out, where progressive disease and there's no bridging options, and the supply chain challenges. So I think the base assumption is you should plan for a real synthetic control arm, or some way to really define the historical controls that you're going into your study design with. And and really look to the expert community. It's an area that's growing very, very rapidly. Still lots of issues and challenges to be learned in, but just stop putting old infrastructure on top of these innovations, and really you have to think differently. I think, as Dr. Paganoni mentioned, you really need the A team around you to do these, and you kind of need the A+ team even to do them for some specific programs such as cell therapies. So,
You bring up a very important point. I think the statement that you made in terms of looking for that large delta, right? I think that is going to be the key as we come up with new therapies. It's in a way a situation where if you have a improvement that is very small delta, you obviously need to use up a large cohort of patients to prove that it's valuable. So it in a way becomes self-defeating. And if you have something that's truly innovative with a huge delta, you can really almost prove that with a small cohort of patients. And obviously, in the whole concept of, I think, the real world controls are really, I think, catching on, and defining the historical controls is something that I think has worked, at least for myeloma. Now we have at least 3 drugs or 4 drugs which has been approved based on data that have been generated on large cohorts that not necessarily were contemporaneous, but did reflect to some extent the makeup of the study population. But I think as we have multiple competing products that are vying for registration, I think this is also an area for collaboration to create real world contemporaneous control. So we know that only 6% of the patients go on clinical trials. So how do we actually learn from the other 94% of the cancer patients? And I would argue that those are the patients who should really be, the data should be captured in a live manner, that they can actually serve as concurrent controls for multiple single arm trials that are ongoing at the same time.
And this is where I would look to Dr. Cho, actually, some of the work that you're doing to understand the myeloma patient population. I think that's a great example of what good looks like, to understand the standard, what the outcomes, anticipated outcomes are. Dr. Cho?
Yeah, I think that's a very important point. Along with all these innovations and developments in the drug field, the treatment fields, and the clinical trial design field, the data field is exploding. And data is going to be a very important commodity in order to fully realize the potential of these innovative trial designs and so forth. The MMRF has sponsored a couple of projects which have been sort of instrumental in establishing that data footprint that's going to potentially lead to agreed upon synthetic control arms that can be applied across multiple trials and so forth. So the first was COMPASS, which is conceived originally as a genomics project, but it longitudinally followed about 1,000 newly diagnosed multiple myeloma patients with concomitant biomarker sampling and clinical data curation. So sites would enroll patients. They would get a bone marrow biopsy and peripheral blood draws. So there was genomics generated immediately on the tumor samples. And then the other samples were banked, which turned out to be prescient, because now with the advent of immune profiling, we're able to extract high dimensional immune profiling data from those same samples. But equally important, patients were tracked through their journey. So initial therapy, first remission, first relapse, second therapy, second remission, and so on and so on. And so by curating this data, we are able to start to assemble what are the treatment outcomes for this group or that group with this therapy or that therapy. But it turns out 1,000 patients, despite that number, is not enough, because there are certain rare subtypes of multiple myeloma, which constitute 10% or less of the total myeloma population, which are considered, like Dr. Kumar pointed out earlier, functionally high-risk patients, for example. They're a minority, but they are the biggest one of the biggest unmet needs in multiple myeloma. So we need more data. We need more patient population data to inform us and lead to effective deployment of things like synthetic control arms. So we have now embarked on a second major project called CureCloud, which is incorporating some of the things that we just talked about. So this is a direct-to-patient effort where patients can read and sign consent forms online. One of the major components of that is data abstraction. So their electronic health records, they give consent to abstraction of their electronic health records, along with biosampling for genomics and immune profiling and other biomarkers. So this is going to give us a look into what happens out in the field. So these are not patients who necessarily go to an academic medical center, whose data is not going to be dependent upon curation by clinical research staff, but rather by trying to leverage electronic health records to amass that data through central repositories. And we're targeting 5,000 total patients for this project over the next 5 to 7 years. So this is a major undertaking, but it's necessary for, in this disease space, which is relatively rare and where the majority of patients are getting their treatment out in the community, in order to get the kind of data that we can use for those types of applications. Obviously, there's a myriad of applications for these types of data. Obviously, we want to understand the biology of the disease better. So the basic and translational components are, of course, key. But one of the ancillary benefits of doing these major types of data projects is that we could potentially accumulate the kind of information we need to create synthetic control arms to understand comparisons with outcomes in second, third line therapy and so forth.
There's so much trial innovation going on out there, and this is where I would have never, like Dr. Paganoni or Dr. Kumar, this is where maybe we point back to you, David, to say, connect us soon and quick, because it's going to take several years for you guys to publish the results of that, but there's so much to be benefited from these collaborations. So not to put you on the spot here, David, but we really look to you to facilitate, some of these interactions, looking at the landscape as a whole.
No, I appreciate that, and certainly, one of the joys of working in this industry, especially at this time, is the ability to collaborate with folks like you. And I think, what we've been seeing is a lot of innovation by necessity. However, also a lot of innovation that has legs and can stand the test of time, because it addresses, very serious productivity and patient centricity issues in clinical research. As my background is actually in economics, and in addition to epidemiology, but originally economics. And what they teach you in economics is that all change happens on the margins. And that's where all the action is. It's not everything changes at once. It's slowly but surely the value starts to creep up and the cost starts to creep down, and then there's a paradigm shift that happens. And I'm cautiously optimistic that a lot of what we've talked about today, and I think in cell and gene therapy in particular, there are challenges independent of the times we're in now, that the promise of those treatments will help drive the medication, the innovation in developing medication. So, I applaud all of you for your intrepidness, if that's a word. I'll say it is, because it takes bravery to do this. It really does. And it takes asking questions, and it takes working really hard, and it takes collaboration. So I do appreciate all of the time. I'll give anyone a second or two to add anything that they might want to add before we close. No obligation to, but if I just want to make sure that I give anybody the floor who has anything else they want to say before we close. Okay. Well, as you may see as you watch this video, the sun is setting here in Research Triangle Park, North Carolina, and it's also setting on this session for a meeting on the Mesa. Once again, thanks to the panelists, thanks to the organizers, thanks to all of the viewers who we don't know who you are yet. Maybe we'll get to know you at some point in the future. Maybe you'll get to reach out if you have questions. But until then, just thanks for this opportunity on behalf of all of us. Appreciate it.